Today I would like to make you know about the use of RFLP and RAPD in plant breeding and crop improvement. So, these are the two markers in the plant breeding technology. So, recent advances made possible to identify the accurate genetic polymorphism. All these technologies enhanced the discovery of the genetic markers. The genetic markers are heritable differences in the nucleotide sequences of the DNA. Nowadays, there are available different types of the markers like morphological markers and molecular markers. We are discussing only molecular marker, that is DNA marker in this class. So, there are two types of the DNA molecular markers are available. They are non-PCR based approaches and PCR based approaches are there. In non-PCR based approaches, RFLP is there, PCR based approaches, several molecular markers are there, RAPD, AFLP, SSR, SCAR, STMA and SNP etc. are available today. Now coming to the RFLP, that is the RFLP is the restricted fragment length polymorphism, which is developed in 1980s by the Alec Jeffrey. So, it is based on Southern blotting technique. It is used to analyze the variations in the length of the fragments that is developed from the DNA shearing. So, to watch the variations in the DNA, the restriction endonucleases are used. The same endonuclease will be used for the different organisms, but the restriction endonuclease makes the cuts at restriction sites. So, the restriction endonucleases and restriction sites plays an important principle in the RFLP, that is restricted fragment length polymorphism. It detects the differential movement of the bond on the gel lanes. So, it is the first molecular technique developed based on the hybridization. So, plants are able to synthesize their DNA or amplify their DNA with high accuracy and rapidly, but at the same time mechanisms are also available which can make the changes and they are operative at the same time. Simple base sequences or sometimes large scale changes like deletions, inversions and translocations or transpositions may occur in the genetic material. All these lead to the gain or loss of the restriction sites, which is leading to the restricted fragments of the genetic material, that is restricted fragments of the different length. These restricted fragments of the different length can be detected on the Southern blots using a suitable probe. So, they are actually detected by the differential movement of the bond, that is band on the agarose, that is on the gel lanes collected from the different strains or species. The main steps involved in the RFLP technique is isolation of the DNA. After isolation, we have to cut the DNA using the endonucleases. Later, we separate those DNA fragments by using the gel electrophoresis. Then they are transferred to the nylon or nitrocellulose paper, which is the material generally used in the Southern blotting technique. Afterwards, visualization of this special DNA can be done by the radioactively probed, that is radioactively labeled probes. So, finally, the analysis of the results are done. So, based on the principle, that is the restriction endonuclease principle, that is which makes the cuts at the restriction sites and makes the different length of the fragments. Because these restriction sites for a particular enzymes are present throughout the genome at several points. That is why the different length of the fragments are obtaining in the DNA digestion. This is the principle of the main principle of the RFLP while doing the technique. Next is that is this is the here RFLP technique, where you are observing the different types of the bonds. that is in the normal case you are watching the 2 bands, whereas in the disease case it is a special band which is displaying in the southern blot diagram. So, after gel electrophoresis, when it is radioactively labeled probe that is detecting the gel that is the RFLP band, then one more band is there in the diseased causing strain. The RFLP technique is here is also one more diagram for you where the whole technique is running, and coming to the applications of the RFLP, we are able to isolate and identify the any type of the gene, and it will be linked to the RFLP locus. What is RFLP locus means? Each band, that is RFLP band developed in the RFLP technique, is called as the RFLP locus. So, these genes would be linked to the RFLP locus. It is a very simple technique and it does not require any sequence specific information. It is a codominant marker, that is why it is used in the differentiation of the heterozygous and homozygous individual. So, it is a supplement to the regular plant breeder as a indirect selection, and it is also used in the detection of the diseases. Coming to the limitations, large amounts of the highly pure DNA is required for the RFLP technique. It needs the good supply of the probes. So, with probes only reliably detecting the variation, that is why we have to give good supply of the probes. Without the good supply of the probes, there will not be detection of variations. That is why there is a need for the good supply of the probes. It is a laborious and expensive process because it is difficult to identify a suitable marker for the genomic DNA or cDNAs. It is a time consuming because it is not amenable to automation. It requires lot of expertism because it is there is a autoradiography procedure where there is a use of radioactively labeled process. So, that is why the RFLP, there are applications that is advantages are also there and limitations are also there. However, it is the one of the efficient protocol to detect the polymorphism. Mainly it is used for the analysis of the genetic diversity of the populations or related species. That is why the RFLP is variously used by the several plant breeders. The next technique is RAPD, that is restricted amplified polymorphic DNA. This DNA marker is also useful in the plant breeding. So, these markers, this technique is actually utilizes the single short oligonucleotide primers of the 10 nucleotide base sequences for the RAPD technique for the amplification of the set of DNAs which are randomly distributed throughout the genome. That is why the name itself RAPD means randomly amplified polymorphic DNA. So, these primers are used to amplify the DNA which are present randomly throughout the genome. So, in the amplification process actually generated from a region which is flanked by a 0 base primes, that is 0 base prime region, that is the priming sites, and all the different genomic materials would produce the different amplification products, but the PCR amplified product is generated only one for one individual, not for others. So, this is the technique, that is RAPD polymorphism among the land races of the sorghum, that is OP, that is these are the some of the RAPD primers having the 10 base sequence, and this is the that is gel configuration on the agarose gels. So, this is a RAPD banding pattern comparing with the some ladders taking from the lambda DNA like that, and likewise RFLP, the RAPD also possesses the same steps, that is like isolation of the DNA, selection of the that is cutting of the DNA, selection of the DNA on the agarose gel, but there is a transfer of the fragments on the ethidium bromide. So, ethidium bromide is a dye by which that is the RAPD's amplified products are visualized under UV light. So, there in RFLP we are transferring to nitrocellulose paper, here we are visualizing under UV light. So, the RAPD applications are, these are construction of the genetic maps and helpful in the mapping of traits and analysis of the genetic structure of the population, fingerprinting of the individuals and identification of the somatic hybrids. Finally, it is very useful system for the characterization of genetic resources. There are certain advantages when compared to the RFLP. It is a simple, quick to perform. The RFLP technique requires high amount of DNA, whereas this technique requires only small amount of DNA. It involves no radioactivity, that is why no expertism is also not required. Coming to the limitations, it is unable to distinguish between homozygous and heterozygous. Actually, coming to the inheritance, the RAPD band actually inheriting from parents to offsprings as a dominant marker. That is why if the band is present, it corresponds to the RAPD dominant allele. If the band is absent, it corresponds to the recessive allele. That is why the RAPD is unable to distinguish between heterozygous and homozygous organisms. The RFLP is able to distinguish between RAPD that is homozygous and heterozygous individuals. This is the comparison about the important characteristics. You can go through this slide, and these are the references which you would like to know. You want to further read about the RAPD and RFLP.